Since today is Resurrection Day, what would tell a guy standing outside a cave with healed wounds? I know most of you would say get some sun, but I am thinking about his future longevity. I would tell them the truth about the biggest scam in the longevity world. Everyone knows that exercise is good. Do you know that exercise has a dose-response curve? Did you know weightlifting is beneficial in lowering all cause of mortality in CVD and cancer until you hit approximately 140 minutes a week? So exercise/weight training has a context. Did your doctor tell you this? More than 140 minutes a week, then your risk of death actually increases. So if you do not want to revisit the cave and the shrouds you had on, you might want to take a look at those slides, because the food guru biochemists won't tell you this. Their knowledge is all from the DoD, DOE, and DARPA. When you are a human and have a Ferrari engine in your head that takes 20% of the cardiac output to think, well, you better know that if you want a fat burn and protein burn to heal, you have to see sunrise, otherwise you will be running to GNC stores to buy creatine. Do you believe the phrase, "Praise be to Orwell because he gave humanity the antidote to the mind poison of tyranny"? Big Harma money produces the centralized science it wants. Yes or no? The hallmark feature of deep state scientific programs linked to DoD and DARPA is to refuse to cite anything that runs against their agendas. The whole truth and nothing but the truth, even it offends your ancestors. That is what we tell guys who just came back from death. New podcast with Nick Jacobs on the evolution of man from oxygen and light selection. A quantum evolution. A new story of paramagnetism that led mammals to a photobioelectric method of regeneration and repair. The lesson is simple. One should take no idea for granted. We should challenge everyone, God, evolution, loved ones, colleagues, supervisors, with deep and meaningful questions to the mysteries in life. We tease out why that is needed today for Maha. 2. If you put a pulse ox on the finger below like I do when my patient has Raynaud's, you can teach yourself something about paramagnetism and med-HB and NO biology linked to blue light exposure. Blue light changes the oxidation state of HBO2. Cold immersion brings it on because of the spectra of mtDNA in this patient's too. They are all Warburg shifted. This is why they all have higher BG, insulin levels, and HbA1c. Light controls this. Raynaud syndrome equals you have too much time in light that causes iron to be in the +3 oxidation state, and this changes the photobioelectrics of the lipid membranes. You get the cold response of Y skin due to focal hypoxic. I told everyone about the paramagnetic toggle switch in Vermont in 2018. Not one person asked the question since that slide was made. Bottom left of the last slide. Nick and I laid out the whole story in the pod above. 3. Best way to fix this problem in surgery, when it happens during anesthesia, is to put NIR light on hand, leaving SAW2 SAT in place. If you have a critical care monitor that measures med-HB, you can teach the CRNA some biophysics right there. Most anesthesia folks use warming to reverse it. Problem is heat does not work like NIR does because of the spectra of NO and HB. I cover how I use the oxygen saturations machines in the ICU here to pull people from the grave who hospitalist MDs almost killed with high FiO2 and endotracheal tubes. Same lesson, but higher intensity and shorter duration than we saw in COVID ICUs, where we induced an oxygen holocaust with algorithmic, centralized medicine. Decentralized medicine saved them from a bad fate. 4. What else can flip the paramagnetic toggle switch in humans? mRNA shots. All viri, many bacteria LPS. More specifically, under normal resting conditions of normoxia and sunlight, most cells begin the process of generating energy by metabolizing glucose to pyruvate. Pyruvate is then shuttled into the mitochondria, where it is oxidized by the tricarboxylic acid, TCA, cycle to generate ADP via the electron transport chain. This process is called oxidative phosphorylation and can generate up to 36 ADP per molecule of glucose. Under blue light, this is impossible. As a result, mammals revert to older evolutionary pathways they used at the KT event. A central focus of immunoneuroMetabolism research relates the metabolic signaling changes of cells to their function. Nearly all mammalian cells adapt their metabolism to transition from quiescent states to activated states that allow for more rapid energy production and changes in cellular capacity. This change is always driven by oxygen tensions and surface light the mammal senses from its eye and skin. This is why neuropsin was evolved and specialized in mammals' exteriors post-KT event. A primary example of this phenomenon is known as the Warburg effect. The Warburg effect refers to a metabolic redox shift due to changing oxygen level due to a change in light that alters the oxidation state in heme iron and hemoglobin from +2 to +3. In the +3 oxidation state, mammals cannot use the TCA cycle well, and they must use older aerobic glycolysis like they did post-KT event. This environmental switch in light and oxygen is felt at the mtDNA level first, where N80+ drops and mammals develop a relative pseudohypoxia. These environmental signals move cell towards preferentially using aerobic glycolysis rather than oxidative phosphorylation, OXPHOS, to generate ADP and macromolecules. This mimics the Earth's environment sometime in the GOE event when oxygen and light varied for the first time in evolutionary history. Nick and I spent a lot of time discussing this. At Trichomes 5, what biophysical things should one know about Warburg redox shifts? They create byproducts fast, but are extremely thermodynamically inefficient. This means they induce a high photobioelectric resistance state in the cell. This means the 30 million volt field on the IMM is not well distributed to the cell or to the organ it is in. What happens in mtDNA when light and oxygen are forced to change because the oxidation state of iron is changed by light. During aerobic glycolysis, cells still convert glucose directly to pyruvate, but pyruvate is subsequently fermented to lactate in the cell's cytoplasm, even in the presence of adequate oxygen. Under such conditions, only 2 molecules of ATP are generated per molecule of glucose. Once cytoplasmic lactate is formed, it must be excreted from the cell to prevent toxicity via acidosis. Acidosis affects how every semiconductive protein operates. Acidosis is a log pH scale, so it affects deuterium biology immediately, and this induces big changes in mtDNA by opening UCP2, causing amplification of COX-2, and deuterium pours into the matrix, acting like maple syrup to make sure the cell cannot use TCA or oxygen well. This is the state mammals keep their stem cells in when they are regenerating or growing in utero. Are there adult tissues that use a Warburg metabolism normally? Yes, places where no melanin exists. Fovea and gonads are 2 examples. A classic Warburg metabolism is therefore characterized by increased cellular glucose uptake and increased cellular lactate output. The brain can use lactate quite well, contrary to what classic biochemistry says. Compared to the complete oxidation of glucose in the mitochondria, cells with a Warburg metabolism generate much less ADP over time. However, the rate of glucose metabolism accomplished by production of lactate from glucose during aerobic glycolysis is 10 to 100 times faster. 6. Why do mammalian stem cells prefer a Warburg metabolism? For the same reason the fovea and gonads do. They are constantly having to regenerate their pieces and parts during adult life. Stem cells are quiescent in adult life, but when mtDNA biophotons 200 to 1000 nanometers light turn them on, they use the Warburg metabolism in extreme hypoxia to amplify growth rapidly. This happens in the fetus, and it certainly was useful in the rebound of eutherian mammals and theropod dinosaurs post-KT event. The Warburg redox shift allows for rapid periods of energy production that can fuel a wide range of biological process, including macromolecular synthesis. This is critical for the fetus, fovea, gonads making sperm, and stem cells of animals with high mitochondrial capacity. For example, rapid ADP production fueled by increased glycolysis can produce metabolites that fuel the pentose phosphate pathway, PPP, and fatty acid synthesis. This leads to the production of amino acids, especially aromatic amino acids that absorb the biophoton spectra that hypoxic mtDNA creates during the event. This also stimulates fatty acids creation that support numerous cellular activities, such as cell growth and division. UV light stimulates mitosis in ultraweak UV BP format. This is all life forms in a photomultiplier showing their endogenous light production. 7. The ability to shift in and out of glycolysis and/or a Warburg-like redox shift mimics what mammals faced coming into and out of the KT era. Light and oxygen are quantized, so it forces metabolism to react. This occurs in response to the energetic demands of light and oxygen and determines the physiological nutritional needs of differentiated organs and tissues. See Nick Lane's slide below. Nothing is special about biochemical pathways. He has rapidly adapted his own opinion over 20 years. The slide shows the adaptation. It also underlies the functioning of most healthy mammalian immune cells, allowing them to respond to infection and other environmental insults. For example, myeloid cells in our bone marrow and liver primarily use glycolysis as a source of ADP. This links to the story I told Nick about Becker's work. These include neutrophils, who are very short-lived granulocytes whose primary function is to rapidly enter sites of infection to initiate microbial killing. They liberate massive amounts of light using free radical chemicals. In fact, B cells shift towards a Warburg-like metabolism to activate antigen receptor signaling. This is why so many people today under blue light are developing autoimmune issues like psoriasis. Nick eloquently spoke about his own AI skin condition in the podcast. Subsets of effector T cells switch to a Warburg-like state upon activation by antigen-presenting cells. Further examples of Warburg-dependent immune cell processes include Th17 polarization by activated T cells, IL-1 beta production by macrophages, and cytokine receptor activation of macrophages. Light, not food, control the game of life. 8. Peter Attia remains limped to explain why PAD is always linked to neurodegeneration and heart disease. The answer is simple. In atherosclerosis, macrophage and endothelial cells in arterial plaque often display increased glycolysis and an inflammatory phenotype. They are all Warburg shifted by blue light, and this keeps arteries with a +3 oxidation state. In the distal organs, they fail as a result. The brain is a TCA-dominated organ, so it undergoes atrophy, and in the heart it is a hypoxically run muscle, and it gets larger to compensate for the energy loss from a lack of TCA and oxygen use. As a result, in both cases, the vascular tree that feeds each organ loses NO production over time and narrows with calcium replacement and loses its local ability to vasodilate to control the CMRO2 of each organ. The effect is clear in the picture. 9. In sarcoidosis, another autoimmune condition, alveolar macrophage and monocytes in granuloma can harbor a sustained Warburg-like metabolism that contributes disease progression due to the biophoton spectra they emit. Recent research fully supports my position that centralized science is a paradigm of pseudoscientific beliefs. Why? Many review articles on the Warburg effect in cancer and atherosclerosis assume that impacted host cells proliferate in a sterile atmosphere. That idea is a joke when you consider our tissues are filled with a stowaway bacterial archaeal hybrid called a mitochondria. It is our original infection we absorbed and usurped to eukaryotic biology. Moreover, the human body is increasingly understood to harbor a tremendous number of bacteria, viruses, fungi, and archaea in tissue and blood, especially under conditions of disease. For example, Kowarski et al. Used cell-free DNA sequencing to identify over 3,000 previously unidentified viruses, bacteria, and fungi in human blood samples obtained from immunocompromised patients. The team concluded that the newly discovered microbes and viruses may prove to be the cause of acute or chronic diseases that, to date, have unknown etiology. Organisms like those identified by Kowarski et al. Often persist in polymicrobial communities that harbor a range of pathobionts capable of changing their gene expression towards pathogenicity and intracellular persistence under conditions of imbalance and immunosuppression. Every one of these infections alters the biophoton emission of mtDNA. This is why these stowaways can cause phenotypic diseases, as laid out by the brilliant Doug Wallace. When energy thermodynamics varies, diseases show up out of the ether. 10. Some sites to support my beliefs above. A. Old Luhuis Elam, Mangul S, Ori APS, Jospin G, Kaslicki D, Yang HT, et al. Transcriptome analysis in whole blood reveals increased microbial diversity in schizophrenia. Transl Psychiatry. 2018, 8/196.
Bevitle I, Leonard Montag, Harrison R, Gan TV, Tonge DP.
Multi-method characterization of the human circulating microbiome. Front Microbial. 2019, 9:3266. C. Connie P.D. Van Hol M. Microbial signatures in metabolic tissues. A novel paradigm for obesity and diabetes.
Ned Metap.
2020, 2/3211-2. D. Kowarski M. Comuna Solar J. Kurdish M. Dovlamenkai, Co W. Pan W. Et al. Numerous uncharacterized and highly divergent microbes which colonize humans are revealed by circulating cell-free DNA. Proc Natal Academic Sci USA. 2017, 114, 3696-23-8. E. Hornef M. Pathogens, commensal symbionts, and pathobionts. Discovery and functional effects on the host. ILARJ, 2015, 56, 2159-62. DOI, 10.1093/ILR/ILV007F Sekner EL. Inflammatory disease caused by intestinal pathobionts. Kuropin microbial. 2017, 35:64-9. Am I nuts, or have I done a deeper dive then, and centralized experts you favors? I let you decide. 11. Adipose tissue cells can also favor a Warburg-like metabolism. For example, Diedrich et al. Demonstrated that bone marrow adipocytes promote a Warburg phenotype in metastatic prostate tumors via HIF-1 alpha activation. My pinned tweet on X talks about how HIF-1 alpha links to PER clock genes. You feeling me yet? What does HIF stand for? Hypoxia-induced factor. We're back to the oxygen and GOE story again, aren't we? Am I wrong, or are they? This should raise the possibility in poor centralized PhDs heads that Warburg-inducing pathogens or pathogens capable of inducing related pathological metabolic states may actually contribute to obesity and diabetes when their electrical resistance is destroyed in those areas and allows direct invasion into the injury bed because regeneration a la Becker's mechanism is destroyed by our modern life.
Where do we live now?
Under light that keeps our iron in a +2 oxidation state or a +3 oxidation state. 12. I'll ask again. Where do we live now? Under light that keeps our iron in a +2 oxidation state or a +3 oxidation state. Is this why this slide below exists? Is this why diseases are showing up because surface-level light penetrates our skin to change hemoglobin's paramagnetic signal, and that information is sent right to mitochondrial DNA, which reacts immediately by lowering NAD+ and oxygen levels and shifting us to aerobic metabolism of Warburg?
Yep.
That is exactly what your modern world does to all of us. 13. I think when you think about this thread and listen to me and Nick has it out, who think is the crazy motherfucker might change rapidly. 14. Soon you'll see all the math I used to figure this out biophysically. I am not here to play with your biochemical experts. I am here to destroy their paradigm of belief and help my species return to being great again. Maha is not the answer. 15. This is Sunlight is Tina. Tina full form. There is no alternative.
16.
I'd like to thank Trichomes for sitting down with me and hashing this out. I hope you enjoyed the lesson.